Good morning, everyone. Thank you for joining today's webinar. My name is DJ Shin, and I am a Sales and Application Specialist from Tomocube. Today, I'll be talking about our company's innovative holotomographic imaging technique and how it can provide new solutions in various different fields of research. Conventional microscopy techniques include bright field, phase contrast, and 2D and 3D fluorescence microscopy. Even though these are easy to use, they normally require some form of fixation, staining, or introduction of exogenous labels and dyes, and doing so can kill the cells or induce phototoxicity and photobleaching. Furthermore, because these techniques use optical illumination, the resolution is limited to 200 to 300 nanometer spatial resolution, and temporal resolution is limited by low signal-to-noise ratio and the long time it takes to scan or illuminate a 2D field of view. Then there are imaging systems like electron microscopy or AFM that provide very high spatial resolution and also quantitative information. However, they take a long time to image and the cells are killed in the preparation process, so they are not appropriate to measure the dynamics of live cells. Also may be quite expensive and difficult to use. We at Tomocube discovered that holotomography can overcome these limitations and enable label-free 3D imaging of live cells and tissues while providing quantitative parameters. You can think of holotomography as the optical version of the X-ray CT. A CT scan measures multiple 2D X-ray images of functional illumination angles. Then, from those multiple 2D images, a three-dimensional tomogram of the body can be retrieved. By doing this, the clinician can look at the organs of the patient without invasive procedures. Just as a CT scan uses X-ray absorptivity as the imaging contrast, HT uses the refractive index distribution of the material being imaged. The refractive index determines the speed of light in the medium, so if you have a higher refractive index, the speed of light will decrease and can be used as the optical imaging contrast in HT. In this manner, HT measures multiple 2D hologram images of live cells, and by scanning the illumination angles and applying the three-dimensional reconstruction algorithm, we can retrieve 3D tomograms of live cells. What differentiates our technology from other conventional 3D HT microscopes is that while other microscopes can utilize rotation and detection using mechanical motors, and they can do three-dimensional scanning, but Tomocube's HT machine uses our patented DMD, which stands for Digital Micromirror Device technology, in order to systematically control the illumination angles without moving parts, so that we can ensure highly stable and ultra-fast measurements of 3D data. So these DMD boards contain tens of thousands of these small micromirrors that can individually tilt and change angles. Light is reflected off of these micromirrors to image around the sample 360 degrees. So using this hardware that we provide and software for reconstruction, it takes about 0.4 seconds for measuring the three-dimensional raw data. And as you can see in the middle, we can retrieve the three-dimensional tomograms of the refractive index. This is a single cell surrounded by high Ri vesicles, which seems to be made of lipid components. And then we can reconstruct the structural information in terms of refractive index and uncover the biological meaning, because there's a linear correlation between Ri and protein concentration. This is how we can provide label-free 3D quantitative imaging, because the refractive index can be interpreted as protein concentration as well as dry mass. This can be done using the software in an automatic manner and also be performed on subcellular organelles, as you can see on the right. The organelles and membrane compartments can be pseudocolored and differentiated based on their refractive index range. And even though we use light as the illumination source, the optical resolution limit should be 220 nanometers, but we can overcome this resolution limit by interpreting the data collected from multiple angles using software algorithms. To reiterate, because we use the refractive index as the imaging contrast, there is no need for labeling or exogenous reagents. Our spatial resolution is up to 110 nanometers because the three-dimensional reconstruction is the sum of the high diffraction angles, and we can apply what we call the synthetic aperture effect. So our spatial resolution goes beyond the diffraction limit determined by objective lens and the numerical aperture. Furthermore, we use a low-energy green laser, and light transfer and absorption by the sample is negligible. So most of the light just transmits through the cell. Since there's very little energy transferred from the light to the cell, you can ignore the heat generation or damage caused by the laser. It takes about 0.1 to 0.4 seconds to measure the three-dimensional data, followed by the reconstruction process, so you can address the very fast dynamics of live cells and tissues. And lastly, From the measured 3D data, you can perform quantification of the acquired image, such as cell volume, surface area, dry mass, and protein concentration, and present them in real physical numbers. Now, I would like to show you what HT has been used for. Using this machine, many researchers have obtained some exciting images of moving cells and tissues. For example, we just introduced some representative data, such as this primary cultured neuron cell. We can see the cell body and axons and dendrites. And since we can measure the three-dimensional images without introducing invasive procedures, we can track the growth and movement of the cells as long as our incubation systems can support, which is 72 hours and over. The live cell imaging capability of HT is well suited for neuroscience as well as cancer cell biology because you don't want to modify or induce different physiological changes to the cell. Unlike the conventional fluorescence microscope, if you introduce fluorescence dyes and staining, you have to worry about if the cells are alive or not, or if they are not healthy anymore, and you have to discard the sample or images after so and so many acquisitions. Then you have to stain the samples again for other measurements. Due to our high resolution and 3D label-free imaging capability, we can also provide clear and high content information of bacterial growth. What we have measured here is a single cell, and the cell has been measured over time, and you can see how they grow and divide and finally form a bacterial biofilm. Like this, there have been many interesting applications from cell biology to hematology to biophysics. In general, HT is well suited for most commercial cell lines, for example, cancer cells. Here we see an example of how our Tomocube's HT technology, in collaboration with our artificial intelligence team, can accurately segment cells and organelles based on not only refractive index, but also AI deep learning. We can then go on to quantify these cells and organelles and observe them in before and after drug treatment studies. Also, you may be wondering, what if you wanted to measure the molecular specific information addressed by fluorescence microscopy and also want correlative imaging for certain information? We can also image HT with correlative fluorescence, so we can capture the hologram as well as the multi-channel, 3-channel fluorescence data. Although using fluorescence seems like it defeats the purpose of label-free HT, fluorescence can provide specificity while HT provides the quantitative and structural information, so they produce a synergistic effect as each provides complementary information. Many of our users use our microscope this way, especially for time-lapse imaging, where if they keep using fluorescence, the cells would die within a few hours. But with label-free holotomography, you can go on for days and days and just use fluorescence once or twice in between. We are not limited to a single field of view, but can also image large areas that go beyond our 80 by 80 micron field of view using our stitching functionality. For example, this is an image of a mouse lung tissue transfected with two different oncogenes, one expressing green fluorescence and the other red fluorescence. As I said, correlative imaging with fluorescence and HT can provide that specificity, and HT's quantification feature can be used to measure and compare the information of each region. We can see in HT and also in fluorescence or with both combined. Normally, if you want to see in fluorescence, you could use confocal microscopy, but to see it together without fluorescence, previously you would have had to stain the sample with H&E. One of the major applications that we can do really well is imaging of lipid droplets. Here in this paper, we observed and compared without labeling the changing of regular macrophages into foam cells, which are macrophages that have ingested lipids. Previous techniques required the use of lipophilic dyes such as Nile red or oil red O to stain and visualize for lipid droplets, but this would incur problems such as the need for fixation, non-specific dyeing, and photobleaching and phototoxicity. However, with HT, we can image live cells and do not require fixation, and we can segment and quantify the lipid droplets with high specificity based on a specific index, refractive index range. From this paper, there was a high sensitivity correspondence of 96% with the HT imaged only and the lipid-specific fluorescence probe. Another upcoming research application for HT is LLPS, liquid-liquid phase separation. These are some data from Seoul National University on protein droplet dynamics. Here we see protein droplets forming as they aggregate, confirmed with fluorescence. And if we visualize with HT only, we can see that the vesicles can be clearly distinguished inside the cells by their high refractive index signal. This process can be captured label-free in live cells, and the changes in number of these individual particles can be quantified over time using our software. Also, just recently, a group from Princeton also used our machine for the study of the relationship between phase separation and cancer metastasis. Immune cells or white blood cells can also be imaged, and one of the projects that we are currently working on with our AI team is the classification of these monocytes. Because we take multiple images in a single acquisition and acquire a lot of data, based on the quantitative biophysical parameters, we can identify the class of white blood cells and tell whether they are B cells or T cells and which markers they have. Also, we can measure the unlabeled cells in 3D and in 3D time-lapse, and we can see how this target cancer cell is being killed by the CAR T cell immunotherapy. Interesting things to note are that only the cancer cell has the high refractive index material as seen in red, while the CAR T cells did not. And we can tell that the cells themselves have dynamics because in a microfluidic chip with flow, some of the CAR T cells actually move in reverse against the flow direction. And because this is viewable in 3D, we can tell whether the interactions or appendages are formed at the top or bottom or on the sides. Like in the previous video, we have measured the interaction between CAR T cell and target cancer B cell. So not only can you see how they form the immunological synapse, but HT can also be used to quantify the synapse area. For example, the kinetic analysis of the cell-to-cell interaction shows the immunological synapse as a 2D synapse area. And also, the protein density can be calculated from refractive index values on the membrane surface. When cells form a synapse, we can see there is a dramatic increase of the membrane protein density in that area. Another important application is with red blood cells, and I think there are many researchers in Singapore who are doing malaria research. Because we can image very quickly without pre-preparation or labeling of blood samples, we can see blood cells and the malaria-causing Plasmodium-infected RBCs. Previously, it would have taken at least 40 minutes and some costly reagents to do GMs staining, and the eyes of a trained expert for diagnosis of malaria. But with HT, we can see any anomalies within the cell in just a few minutes. Also, our CTO, Professor Paul Park, recently did a platelets webinar. As you may know, these are important cells in the blood system, which provide a crucial role in collagen and blood clotting formation. Before activation, the size and shape of these platelets are only 1 to 2 micrometers in diameter. So with previous imaging techniques, it was challenging because to introduce contrast, you would have to alter or kill the cells, which makes it impossible to capture the fast dynamics of these small cells. Also, we can see that the structure is actually heterogeneous, and there are regions of different higher refractive index in the pink inside the cells. So things like this and also the fast platelet activation process all have not been seen or captured before. For microbiology, classification has become much easier and more accurate with HT. While previously we would have had to use PCR to identify species of bacteria in a sample, now with HT and AI we can do it within 1 minute of imaging and with high accuracy. If you look at the graph, you can see that we were able to identify multiple bacterial strains with 94.5% accuracy. But this was in 2017, I believe. Now it is at 97% accuracy. This is important for cases of sepsis, for example, where toxins from bacteria can incur septic shock to a patient. If a patient has been diagnosed, time is the most important factor to identify which bacteria is in the blood. From my understanding, the time it takes for current tests to get results is about a day or two, and a person could die in that time if they are not treated. Our bacterial cell counter differs from other commercial cell counters because we can image in 3D and tell if the bacteria is at the top or bottom of various layers of biofilm, leading to more accurate analysis. We are also getting some very interesting results from algae studies using our technology. For example, in one case, it is now easier to identify a certain algae species in river water based on their morphology, because we can see the 3D and cross-sectional images of each refractive index distribution. The starch and chloroplast content inside can also be quantified. The structure of diatoms and their different subcellular organelles can also be easily identified, label-free, and quantified for refractive index, volume, and mass. Also, sometimes it is very difficult to capture the very fast dynamics of dynamic samples such as sperm or certain parasites like this trypanosoma, but we could capture them in time-lapse with our 0.4 second acquisition speed. For nanoparticles, the author of this research publication was developing an anti-cancer drug that was bound by a gold nanoshell. He wanted to load this inside a macrophage for drug delivery. The 3D images in 3 directions of view show that the gold nanoparticles are loaded well into the macrophages, and we observe this process by 3D time-lapse. Red indicates the positions of the gold nanoparticles, and blue shows the cell membrane. In the center of the cell, we see that the color becomes a darker blue due to the thickness of the cell membrane. This shows that the ex vivo fluorescence image and quantification analysis would be a potential bioimaging technique to validate the biodistribution of the gold nanoparticle loaded macrophages in a nanodrug delivery system for cancer therapy. Refractive index inside the spores can show which ones have higher density inside, meaning more nutrition, and which ones are more likely to mature and grow well. Measuring the 3D structure of individual pollen grains remains challenging using conventional imaging approaches, mainly due to the lack of capacity for quantitative 3D measurement. And even advanced 3D imaging methods have limitations or may require highly specialized equipment. And that concludes my presentation for today. Again, thank you for joining today's webinar. If you have any questions about Tomocube or holotomography, or would like to see the presentation materials, please feel free to email me anytime at djshin@tomocube.com. Again, thank you and stay safe, everyone. See you next time.